So I asked Sarah to come and give us a primer on that. Sarah is actually a postdoc in my husband's lab. And I know that she does things meticulously. Her experiments are amazing. So I thought she'd be the perfect person to come join us. She did her Ph.D. in Germany, and she worked on chromatin dynamics and gene regulation, and developed a very strong interest in the emerging field at that time, right?
Yeah, at that time.
Sequencing technologies, including RNA-Seq. And so she took the good route, unlike me. She did her PhD by combining wet lab and bioinformatics, which I think is a very clever use of time. I didn't learn bioinformatics, so I always had to pay somebody to help me do that part. And about a year ago, she joined my husband's lab, as I mentioned, and she started working on long, non-coding RNAs, which David's lab had been working on for several years and found some very cool things about them, as opposed to what some other comments may have heard this week from who we won't mention. So long non-coding RNAs do play a role in disease and other types of developmental processes. So she's now looking at the non-coding transcriptome in several breast cancer models, mouse models. And of course, the focus there is on discovering new prognostic and therapeutic targets. So we're not going to talk about her research today, unfortunately. That would take another whole hour at least.
At least.
I asked her just to focus on validating this RNA-Seq data. So Sarah, thank you for coming.
Thank you very much for having me. Thank you, Mona, for the introduction. So one big part of this whole RNA-Seq experiment is obviously validating. And the first question I usually get whenever I present any of my RNA-Seq data, and I have a lot, is, yeah, but how do you know that's true? How do you know that's real? Are you sure that's not just an artifact? You know, these kinds of things. So definitely a major question here is, how reliable is our RNA-Seq data? And what does it really mean? I guess most of you will probably work on differential gene expression. And obviously, you want to know if you find a differentially expressed gene, is it actually differentially expressed? Or is it some kind of technical artifact you get here? I don't know if anyone is interested in different splicing isoforms, but you can also detect novel isoforms by RNA-Seq. And how do you know that those are real? So it's a very tricky question, actually. And there's a lot of different methods to validate them. I'll give you an overview of what these methods are. However, I will focus specifically on qPCR because, again, when I'm giving my talks, usually people are like, but have you done qPCR on these genes? Have you verified those? And this is probably the commonly used method, and that's why we will focus on that today. But I'm happy to answer any other questions as well, of course. There's a pretty good paper which I would like to mention, although it's already 4 years old. But they have done a very extensive study of comparing RNA-Seq data to microarray data, to qPCR, to Sanger sequencing, to everything you could think of, and have verified how well these methods correlate. So it's a pretty good starting point if you're interested in anything else but qPCR. Now, what are the different validation methods for RNA-Seq? In principle, you have two options. One is qualitative validation, and the other one is quantitative validation. Now let's start with the qualitative ones. Most obviously, the first one would be reverse transcription PCR, so just a standard cDNA synthesis, and then you amplify your cDNA, and then you run it on a gel. This is, of course, not quantitative. It's just a qualitative approach to see, is it actually expressed? And maybe you can get some glimpse of how strongly it's expressed, but it's really not quantitative, so I wouldn't use it for that. If you are interested in finding new exon-exon junctions, or maybe some SNPs at some stage, you could also use classical Sanger sequencing to verify these kinds of things. And finally, if you would like to see it, if you would actually like to see your transcript in the cells, you can do in situ hybridization like RNA-FISH. And I just quickly want to show you a picture, which is actually from a publication from our lab, the Spectre lab. And you can quite nicely see those red dots are a transcript called MALAT1, and those green dots are a transcript called NEAT1. And this displays already the second advantage of RNA-FISH is not only you can see your transcript and know it's there, you can also tell where it is. So RNA-FISH has, in addition to the qualitative validation, also a localization advantage. Now, there's a number of quantitative methods you can use to validate your RNA-Seq data. And the gold standard is actually still northern blotting, although it's super old. Has been used for many, many years. But it gives you not only actually quantitation, it also gives you different splicing isoforms, for example, and it also is necessary for some specific applications, let's say microRNA sequencing or small RNA sequencing, which you might not be able to amplify very well with PCR, so you actually have to do a northern blot. However, northern blotting is, as I'm sure you all know, technically not super easy. It's very elaborate. You need radioactivity in most cases, which you don't want to use. And most importantly, I'm sure many of you are working with tissues or like just low yield of RNA, and then it's simply not feasible to do a northern blot if you have just a picogram of RNA. So you can also use microarrays. It depends a bit on what you actually want to answer, which question you want to answer. So maybe if you're interested in a range of genes, you could design a custom microarray and just test all your genes on the array. Microarrays are a lot more quantitative than RNA-Seq, in fact. If you're just interested in 1 or 2 or 10, yes?
What did you just say about microarrays being more quantitative than RNA-Seq?
Yeah.
I thought they come to a fluorescence saturation and
It depends a lot on what we are comparing. So if we compare this classical RNA microarray where we just hybridize, just a standard hybridization, the double hybridization, you label your control in green and your sample in red. Yes, they do run into saturation, but actually you have a control on your chip.
So you just choose a control that has
yes, so if you
that's comparable.
Exactly. Of course, there's different types of arrays, and some do not allow hybridization with a control, and those are, of course, not the ones I'm talking about. I'm talking about the double hybridization ones, because then you have a control on the same cell, and they all hybridize, at least statistically, they should hybridize with the same probability to the probes, and then you can quantify a transcript like a qPCR, just a wider range in the end it is. If you just hybridize a control on one chip and then your treatment on another chip, then obviously it's not going to be as quantitative anymore. But I'm talking about the very classical red-green ratio microarrays. And
I still don't see why that measure should be more quantitative than the CT values.
oh no, it's not more quantitative than CT values. No, it's more quantitative than RNA-Seq.
Oh,
So like,
sure.
okay, sorry, maybe I said it wrongly. No, of course it's not more quantitative than qPCR. qPCR is the most quantitative method we have. I just wanted to make the point between RNA-Seq and microarrays. So quantitative real-time PCR is, as I said, the most quantitative option you have. And it is also very commonly used, just if you're looking. Okay, sorry. Can everyone hear me? If you're just interested in one gene or a couple of genes, you just want to verify the most interesting ones from your RNA-Seq screen, then qPCR is perfectly fine. Now, how well do RNA-Seq and qRT-PCR actually correlate with each other? There's a lot of studies out there who compared exactly that, and they correlate reasonably well. That's what we can say. You have here on the x-axis a differential expression as measured by qPCR, and on the y-axis the differential expression as measured by FPKMs by RNA-Seq. And you get reasonably well correlation. So the validation rate in this particular case was 88%. And this is also what you more or less get. So between 80 and 90% is what you usually should get between RNA-Seq and qPCR. The blue dots here are all the validated targets. Purple ones are the so-called housekeeping controls. And then there are some red dots who are not validated, which means they did not show up differentially expressed in the qPCR, but they did show up differentially expressed in the RNA-Seq. And usually that's also what you get. So that's a classical example. It happens sometimes that a gene shows up differentially expressed in your RNA-Seq screen, but you might not be able to validate it afterwards. So
Can I ask you a question on this? Is there any correlation with the level of gene expression? So
Yes,
small,
yes.
like low expression
Very good
correlation?
question, and you're absolutely right, there is. And usually all these correlations are best when you are in a medium range of expression. Very highly expressed and very lowly expressed genes have some technical issues in both methods, RNA-Seq and qRT-PCR, and that's why the correlation is diverging both up and down. So the medium range, like the standard protein gene expressions, that's where the correlation is the best.
Medium, can you put like specific
Okay, approximately, I would say if you're looking for protein coding genes, for example, I would probably not go for anything which is expressed lower than 10 FPKMs, for example, because that's usually, it's oftentimes considered as noise. It's background. You don't really know, is this a lowly expressed gene? Is this a lowly abundant gene? Or is it actually noise? So it's hard to give values, but I personally wouldn't go for anything below 10 FPKMs. I don't know if that helps.
What's the high? Where you start having
Thousands.
Okay. So, I mean, I know, yeah, so that's where it really starts being a problem, if you like those really crazy highly expressed genes.
Yeah.
Is there any correlation between the Q value associated with the log difference and the validation? It looks to me like most of your red dots fall within about,
These ones?
one-fold or two-fold.
Yeah, so the cutoff line here would be by 1, and this is log 2-fold, so it means these are 2-fold or more overexpressed as determined by RNA-Seq. However, they could not be found in the qPCR. I don't actually know if there is there's any statistical correlation and you say, oh, if my Q value is basically 0, then for sure this is actually a differential expression or not. I guess the best way to find that out is by doing biological replicates and see if you see the same effect more often. Then I would rather believe in it than if I just see it once. And I wouldn't be too hung up on the statistics in that case. Okay, now let's get technical. In general, you have 2 different options for performing your qRT-PCR. It's either 1-step or a 2-step qPCR, and depending on what you're trying to do, one or the other method might be more beneficial. Basically what it means, in a 1-step RT-qPCR, you do both the cDNA synthesis and the qPCR in the same tube. So you have your RNA, your reverse transcriptase, a DNA polymerase attack, and of course buffer and dNTPs, and you only have sequence-specific primers. So you add your PCR primers right to the cDNA synthesis, which has the benefit that only those get amplified, and you have a very sensitive measurement in that case. However, you're not very flexible because you don't have any cDNA which you can use afterwards. If you want to run a different gene, a different target afterwards, you basically have to do the same thing all over again. So it depends if you want to verify loads of targets with the same cDNA, maybe you want to go for 2-step. If you want to just really check one, but you have a panel of 96 different ones on a plate, let's say if you're doing a screen, it might be a bit faster. But it's really up to you which one you prefer. 2-step qPCR would be just as I laid out. You have a RT, a cDNA synthesis step beforehand with either oligo-DTs, random primers, or you can also here use sequence-specific primers. And then you use the cDNA subsequently for a classical qPCR. Mm-hmm.
Sorry. Can you multiplex the 2-step when you have the...
You can multiplex the 2-step. You can not multiplex the 1-step. So depending on your application, either the one or the other might be beneficial.
So how far can you multiplex?
It depends on your qPCR machine. So depending on how many filters it has. So in my old lab, we had one that could do 6 different colors.
I guess I'm asking specifically about the RT reaction. If you're going to do
About the RT reaction?
Right. So if you're using sequence-specific primers, I may...
Oh, okay. Okay, I see.
B, C, D, E. How far can you go?
Yeah, I guess I would actually then not multiplex it. I would prepare one individual reaction. That's what I would do, just to make sure.
We've tried with 2 and it works, but more than that.
More not.
Yeah,
Okay.
and then you have the heating amplicon.
So if you're going the 2-step way, what do you recommend? Random primers? I mean, we use random primers.
Yeah, so again, it depends on your application. I personally use random primers because I'm working on non-coding RNAs, so I want everything to be amplified. So random hexamers, of course, amplify your rRNAs, your tRNAs, everything which doesn't have a poly-A tail, whereas oligo-DT only amplifies things which have a poly-A tail. So if you're only interested in those transcripts, maybe oligo-DT is the better option, especially because then you can be sure you're amplifying a full-length transcript, whereas with random hexamers, you also amplify everything which has been degraded and is only present in fragments anymore. Okay, now how does this whole process of qRT-PCR work? This is a typical qPCR curve. You have in the beginning an exponential phase, which means at this stage neither your primers or your NTPs or anything else in your reaction is limiting, and that's why you actually have an exponential growth. So out of, let's say, 1 nanogram of cDNA, you get in one cycle 2 nanograms, and then you get 4, and it's just growing exponentially. However, very soon you're entering the linear range, where then primers and all other components in your reaction will become rate limiting until finally you reach a plateau, which just basically means that your detector at your qPCR machine is saturated and can't detect any additional fluorescence anymore. And if you have a good qPCR reaction, that's how you want it to look. So you want it to actually plateau out. If it just ends here around it, maybe you want to use more RNA for your cDNA synthesis or have a higher concentrated cDNA in your reaction. There's a lot of troubleshooting you can do, but in the optimal case, it doesn't work in all cases. Okay, granted, but in the optimal case, you reach a plateau here. And there's a relatively arbitrary set threshold, which is just defining what is actually a product here and what's noise, what's background. And as soon as your product is reaching this threshold, this is called the cycle where it reaches this threshold, is CT value, and that's what you actually use for calculations afterwards. This would be a dilution row just to show how this would work. If you have 10 picogram of starting material, your CT value might be relatively late, maybe at 35. If you have 100 picograms, it's coming up already cycles earlier, and then 1 nanograms, 10 nanograms, 100, and 1,000. So you just can see that it is very dependent on the concentration, and in the optimal case, it should be a linear correlation if you plot the total RNA concentrations versus your CT values. That's kind of a quality control. So you want, if you get this plot, you want that the R square, the correlation coefficient, is very close to 1, so 0.99, 0.98, something like that. And you just want to have a nice linear correlation here. This is just a quality control to see if your reaction works at all or not.
So I thought we want the slope to be like minus 3. There's some number, I can't remember what it is.
Minus 3.32 or something?
Yeah.
It is.
The ones where our correlation is beautiful and our slope is
Yeah.
so I never.
So this has to do with primer efficiency, and I will actually cover that later on. So can I postpone this to later, or?
That'd be great.
Okay, fantastic. There's a lot of different ways of performing qRT-PCR, and that depends on your system, on whether or not you have a Roche light cycler or Applied Biosystems or whatever you have in your labs. They have different technologies. I guess the most commonly used, apart from Cybergreen, would be Tagman chemistry. I will only cover Cybergreen in the interest of time, and I only know about Tagman otherwise, so please don't ask me about molecular beacons or something like that. I don't know it. So Cybergreen is the commonly used fluorescent dye for a qRT-PCR, and the good thing about it is that it has almost no fluorescence when it's not bound to any DNA, and as soon as the primer anneals, also Cybergreen can intercalate. It intercalates only in double-stranded DNA, and then in the course of amplification, you get more and more Cybergreen intercalating, and thereby more and more fluorescence, and that's directly correlated to the amount of cDNA, as long as you are not exceeding the exponential phase or the linear range. Okay, now I was advised to give an introduction into primer design, so that's why I'm putting a lot of time into that now. I hope that's all right. Please, if it's all totally obvious, let me know. So there's a couple of general guidelines on how to design the best optimal qRT-PCR primers, if there is something like that. And if you look it up online, you'll get thousands of advices, like how many G's and C's should be at the 5' and at the 3' end and whatnot. I personally go with these criteria, and they have always worked fine for me. So I guess in 99% of all cases, you will be fine and not run into any problems. Now, the most obvious criteria obviously is it should be unique for your target sequence. So unless you're working with a pseudogene or anything which is very repetitive, you shouldn't have any problems, but you still should verify that your primers are actually specific only for your target. And I'll come to specificity in a bit. Secondly, it's beneficial if the 2 primers you design are not prone to primer-dimer formation. Otherwise, it could easily destroy your reaction. And also, they should not form any secondary structures if possible, because again, annealing will be inhibited. Usually the recommendation is there should be between 18 and 25 base pairs. That's enough to get them specific for the target in the most cases. And they should have, both formatted reverse primers should have a comparable GC content, because that ultimately results in a comparable melting temperature. And of course you don't want one primer have a melting temperature of 50 and the other one of 80. That's not going to work. An optimal range of melting temperature is usually said to be about 60 degrees, but in my experience, anything between 55 and 65 works fine. The length of the amplicon, again, you will find a lot of different advice here. For me, 60 to 200 has always worked, and I think it's a very common range to use. And finally, if you can, then your primer should span exon-exon junctions. And here's why. Even if you treat your cDNA reaction with DNAs or your RNA extraction with DNAs, and I guess many actually don't do that to start with, you might have some contamination of genomic DNA in your sample. And now if you have primers who span exon-exon junctions like this one does, so half of the primer is in exon 1 and the other half binds to exon 2, it can not amplify genomic DNA, but it can perfectly fine amplify cDNA. Now, the same is true if you have a very long intron in between. In case you can't design anything which borders exon-exon junctions, you can choose some which are separated by a very long intron. In that case, it's also very unlikely that any product will appear from genomic DNA. I have a list of thousands of databases which you could use. And of course, you all get those in the end, so you don't have to copy my slide here. There is a number of non-commercial ones, which means they are free, and that's what I always recommend to use. I have pretty good experience with Primer Bank from Harvard University. They have a very extensive set of qRT-PCR primers, and the good thing is you usually also have a gel picture of those primers, so they actually run it on a gel, show you the picture, and then you can decide whether or not this is according to your standards. So sometimes you look at primer sequences, you see the gel, and there is no product. I wouldn't order those, for example. It happened to me, I promise.
So that's not a design database, that's just a database of known
Genes.
Yeah, exactly.
But they're all like mouse, human.
Yeah, so that's exactly what I wanted to mention. Most of those public databases are for mouse or human because they're just the most commonly used organisms. Some of them are also for other organisms, but if you are working with something very exotic, you might not be successful with the public databases. There's a lot of commercial ones as well, and if you're working with applied biosystems, you might consider that you actually take some primers which they have validated for you. They do cost money, but at least they will work for sure too. So.
Do you have a favorite, website for primer design? Like I use IDT. Okay.
Yes, I do. I guess the most famous one is Primer3. And I have only one web page here, but it's open source, so many other universities, there's definitely a link at MIT and then probably at any university everywhere, they're all using Primer3. And it is pretty good. It is. These are free, completely free, yeah. So Primer3 is open source, which means it's web-based. You just copy-paste your sequence there. Mm-hmm.
I will say one thing with Primer3, check the TMs, because when I order things from IDT, they're not the same as with Primer3.
It depends how it's calculated.
Yeah, so it is true that if you design anything online, they will give you an estimated TM, and then you probably order the primers and you get a different TM. That's true. They shouldn't vary too much, at least. They can vary by 1 or 2 degrees. 5, really? Okay, that didn't happen to me yet, but okay. I usually I try to design my primers around 60 degrees, even if they say it's 59, I still run them with 60. It has always worked so far. But of course, something to keep in mind that whatever they predict as melting temperature might not be the actual melting temperature. And I'll come to that too later. There is also a web-based surface called PrimerBlast, and PrimerBlast is NCBI based. It just combines Primer3 software with a blast search. So that's also quite handy. I use it a lot because I'm working a lot with pseudogenes, so I need very specific primers, and you just copy-paste your sequence there. You have to adjust the length of the amplicon because by default it's, I think, 100 to 1,000, and that's too long for qPCRs at least. But otherwise it's really good, and it will give you not only primers that work work, but also primers that are specific to your transcript right away. There is an open source software called Benchling, which can be used also for cloning, and I think maybe nowadays even for high throughput sequencing analysis, a little bit at least. So you could have a look at that, it's for free. And then there's something which I have used a lot in the past because it's one of the few softwares which run on a Mac, and that's called Genius. However, that's not free. It is also based on Primer3 though, and it also has all these vector NTI opportunities, like you can do cloning or also high throughput analysis. Actually with this one I know for sure you can do that. Now about primer specificity, even if you do not design your primers with PrimerBlast, it is very beneficial if you afterwards use PrimerBlast or just a basic blast search to verify my primers are actually specific to my target and they don't accidentally bind to 100 other transcripts. You will see that sooner or later, but maybe you want to know that before you order the primers and do the whole reaction. Another point, and that's coming back to the melting temperatures, this is pretty much out of fashion nowadays. No one is optimizing melting temperatures anymore. I have done it a lot actually, definitely in my undergrad and also in my grad work. So you actually could run a gradient if your PCR machine is suitable for a gradient run, of course it is. And try different temperatures from 55 to 65, for example, is a common range, and see if your primer is specific or if it amplifies your target at a certain temperature range better than in another one. Yes?
Going back to the primer blast, looking for unique primer binding sites, it can often be difficult, especially in a genome such as the plant that I work with is internally triplicated. So if you have to use a primer that anneals or the blast score is that 23 out of 24 is the potentially competing site. I mean, there's all sorts of variables, how much mismatch or similarity you can tolerate, and I think it also matters where those mismatches are.
Yeah, definitely. Definitely.
It's less consequential than
exactly. Exactly.
Do you have any guidance?
So usually if I have 4 mismatches, I don't bother because they shouldn't align to. They can maybe in theory align, but honestly they will not in practice. At least I haven't seen that so far. I am running into similar problems with my pseudogenes sometimes because they can be up to 99% sequence homology to the protein coding gene. And what I do then is I actually blast my entire sequence of the gene I'm looking for, not primer blast, but use the actual blast, and then look whatever is closest, and then go through the FASTA alignment and check for SNPs. And I actually check, okay, maybe there's a gap, so this is a good region to design my primers across. Or if there's really just a couple of SNPs, I design both forward and reverse primer on a SNP and try to have them as the last or second last base of my primer on the 3' end, because it still could amplify, and definitely in these cases you should probably use standard Sanger sequencing to verify what you're sequencing here, or use some other kind of verification of what you're amplifying. But it is helping in getting the right target sequence, at least in my experience.
In this case, could you also play with the temperature maybe to try to
You can try.
challenge?
You can definitely try and see if it at 55 degrees amplifies, in my case, the protein coding gene, and at 65 maybe only the pseudogene, because there is a AT to CG base change or something like that. So that's definitely, you can also play with magnesium concentrations. That can also help if you do a gradient like that. But I have the feeling that most people just nowadays order primers, do a qPCR, don't bother too much. So there's still a lot of things you should actually bear in mind, I believe. Another good way, and this is actually also be used for SNPs, is a melting curve. So I would always recommend to run a melting curve after your actually qRT-PCR reaction, because it will tell you how many products you get. And you can do a high resolution melt curve if you're bothered about sequence specificity. So if you have even if you have only one SNP in the entire amplicon, a high resolution melt curve should tell you which one it is, or if you get 1 or 2 products. So that could be an option too. And also essential is always run your primers on a gel. It sounds old-fashioned again, but at least if you have new primers, even if you have a perfect melting curve, you might want to see it on the gel and see this is only one product, and this is certainly not a primer dimer, what I'm seeing here.
But you don't do the two primers. You said run the primers. You mean run the PCR?
Yeah, the PCR product. Yeah, okay, sorry.
But you can't do the melting curve and you can't do those two, right?
I do both.
Yeah,
but not on the same samples.
Oh yeah, you can run afterwards. After the qPCR is finished, you have the plate or your tubes,
Yeah,
and you
but
just run it on a
Melton
gel.
curve, aren't they denatured at this point?
Oh no.
No?
No.
They fine?
You can still run it on the gel. Yeah.
Okay.
Do you ever find it necessary to actually send an amplicon to sequence to verify?
Only in case, as we talked about, the sequence specific effects. If you're not sure if your primer is amplifying a pseudogene or a protein coding gene, something like that, then yes. Otherwise, if you know that your primers are specific and they can only amplify this one transcript, and you have the right band on your gel and your molecule looks fine, I would not send it for sequencing. I don't think that's necessary in that case. And finally, a good way to verify that you're actually getting the right product would be multiple primer pairs per target. And if you get the same result with all these primer pairs, then you for sure know that what you're measuring here. Now I'll show you an example from a publication. This is, by the way, Tom Watson, not James, in case anyone is wondering. And it's about the expression of a visual homeobox gene in mouse, and they compare retina to cornea here. And I'm just showing this because it's a nice example of how a qPCR can look like. They use 3 different primers, which are these 3 blue lines. They are not reaching a plateau, so that's the only downside of this paper, I find. But otherwise, they have a control grade gene. This is GAPDH in that case, which they normalize it to. And they have also a row control, which is specific for the retina and not for the cornea. So that's how they verify their results. And that would be a good example to do it. And it's quite obvious that this VSX protein is only expressed in the retina, and there's absolutely nothing. There's baseline in the cornea. So if you get something like that, you could be reasonably sure of what we're doing here. They also run it on a gel afterwards. The 3 primer pairs, I hope you can see. There's 1 product, 2, and 3. So it's a single band. There's no primer dimers. It looks really clean. And same with the GAPDH and row primers as well.
So I would be a bit disturbed or concerned because the primer pair 2,
This one?
I presume these are amplified to saturation, and yet that band is...
Yeah, actually you can't see it very well on the screen. It looks a lot better on my computer. So I think it's just a matter of display. This is a little weaker, but it's pretty similar, same range as the other 2 are on my computer. I can show you later if you like.
The size difference in so
No, I was just thinking about yield and the efficiency of amplification. So I don't know how that plays into the CT values that you determine. Probably not. I mean, if it's less efficiently amplified.
So that would be primer pair 2, and as you can see, it has a little lower CT as the other 2. So it does affect them, but usually if you run into saturation, in the end, you shouldn't see a difference of your primer pairs on the gel. Okay, now just a comment about melting curves. This is a specific product, so you get nothing, and then at a certain temperature, you get a peak, and then that's it. That's how it should look like. If you get 2 peaks, like 1 here and 1 there, you have probably some primer dimer contamination here. In this case, this is the negative control, and in the negative control, it is fairly common that you do get that because there's nothing to amplify really. So that's okay here, but in your specific product, you really just want 1 single peak. Okay, I have to hurry a bit, I'm sorry. The last thing I want to talk about is how to analyze qRT-PCR data. And most people, I guess, still use the delta-delta CT method. So that's why I'm talking about that one. And I will give a short note about primary efficiencies and other methods in the end. So basically, you need 4 different things. You need a control gene. For basically every target gene, you need always a control gene. But you also need an actual biological control of your treatment. So this could be an untreated case and a treatment case, or 2 different genotypes, or whatever you're working on, basically. So red line here would be the treated target gene, and the red dotted line is the treated control gene, and the blue dotted line is the control gene, and the solid blue line would be the target gene in the control. And you can see that basically the control genes, both in treated and untreated case, are pretty similar, and that's what they should be. So if you have a large variation of your control genes already, you can probably not really quantify anything here. You can see also that there's a bigger difference between the target gene, so actually your treatment seems to have some kind of effect in that case. Now, how do you calculate this exactly? As the name implies, delta delta CT means you have to make two deltas. And the first delta you calculate is the difference between the CT value of your target gene and your control gene. Control gene, again, could be like GAPDH, for example, and the target gene could be this visual X protein we just had in the example. And then you calculate the difference between your target gene and your control gene, and you do this for both your biological control, like the untreated case, and also for your treatment or your condition, whatever you want to compare to each other. So you have two CT values, and they call it calibrator here. So that would be your control or your untreated case, and then your ever unknown condition or your treated ones. And you calculate delta CTs for both, and then you can calculate a value called relative quantitation, RQ. To calibrate, that would be 2 to the power of minus delta delta CT, and delta delta CT is the difference between your calibrator, your untreated control, and your treated control. Does that make any sense?
That's the question, kind of fundamental. How,
Mm-hmm.
You said GAPDH, but like if you're working, how do you choose the control machine?
I'm actually coming to that. Can I postpone it for a few slides? Almost there, no worries. I just want to make a point about primer efficiency because there was a question about the slope. So obviously for the whole delta delta CT method, this just works if you're comparing apples to apples and not apples to peers, which means the primers of your two genes you're taking, your control gene and your target gene, should amplify with the same efficiency. And I agree it is not in all cases very easy to get. You can again calculate this E value where you plot on the y-axis the delta CT and then the x-axis the log of the relative quantity, and then you should get a linear correlation and you get a slope. And now the slope in the optimal case is something minus 3.32, something around that. Basically, it just means that in the end, if you solve this equation, you want that your efficiency is 1 or 100%. And that would then mean you have an exponential increase from 1 nanograms to 2 to 4 and so on. And now most primer pairs don't have 100% efficiency, but you should aim for something between 95 and 105. If you're in that range, you're pretty good, and then you can compare these different ones. If you are not in that range, you have two options. One option is design new primers, and that's what I would always go for. The second option is if for some reason this really doesn't work, you can use an efficiency normalized method. And I can give you a handout where they actually have a paper of where this is described in very detail. And very long story short, it just basically means that instead of 2, you have a different value here because you can't assume that you get the same thing exponentially. So maybe if you have a primer which only amplifies with 90% efficiency, it would be 1.9 to the power of minus delta delta CT. Something like that. It's not the optimal case, but many people do use it too.
So that's on the applied biosystems that describes how to do that?
Oh, there's a whole paper about it, and I'm happy to contribute it. Like if you
Is it the Pfaff paper?
Is it the what?
The Pfaff, I don't know how to say it.
Oh yeah, exactly. It's from Michael Pfaff. I think he sounds German. I'm not sure if he's FF. I think it's PFAFF. Or PFAF. I'll give you the reference anyway, so you can read it up on it if you like.
Did you say what efficiency you're looking for?
I would aim for 95 to 105%. If it's only 80% or something like that, I certainly would not use that primer pair. Mathematically, you can.
Yeah, we've had it. We never figure out how, but
There is an explanation of how I think about it. There is an explanation.
Yeah,
You just have a higher fluorescence value than you should have for double as much product as you. So you would expect that your fluorescent is linear to your increase of product. So for 1 to 2 to 4 nanograms, you also would expect 2 to 4 fluorescent units. So if that's not the case, for example, because your primers form a little bit of primer dimers and you have additional fluorescence, or for any other reason, which happens in your tube, you have too much fluorescence, then you get an efficiency of 105 or even more than that.
The other thing, I mean, just one thing, we've noticed like sometimes we'll get primers from a paper and we use them in our lab and we get funny results and then we go and check these things and they're not, so just don't trust it just because it's in a paper doesn't mean they optimized it. And you could also use slightly different conditions or slightly different
That's a very good point.
reagents, but just be really careful. We've had
That's a
number of
very good
times.
point. I actually stopped using any primers from any publications because they never worked for me. And I always get like triple bands. And it can, of course, depend on different buffer systems, different qPCR machines, different people pipetting, I don't know. But I don't have very good experiences with those either.
Yeah, it's just weird.
Question about if you get primers, if you order primers, you run them at 60 and you don't get a good efficiency, will you run a temp gradient or just order more primers? Try to stick with everything at one temperature.
It depends on how difficult it was to design your primers. If it is a standard protein coding gene and you can design easily a new primer pair, I would go for that. If it's a bit more tricky, I would run a gradient. It's also not every machine can run a gradient, so it depends on what equipment you have in your lab a little bit.
Okay, so do you find though that oftentimes if you get a bad efficiency, you can fix it with running a different?
Actually, yeah. So in my entire PhD, that's what I did. I'd optimized every single one of my primers to the optimal TM, and just use this TM in the future, because then you have maybe 95% in one case, but 100 if you just increase the temperature by a degree. So that's what I did. I'm not saying everyone has to do that, of course, but.
And then to get your efficiencies, you run like how many dilutions and how many replicates do you?
Usually a dilution of 10 samples is what I do, and in triplicates, as everything always in qRT-PCR always triplicates.
Okay.
If there's, unless you have a very good reason why you can't use triplicates.
Thank you.
Sure. Now the last thing I want to talk about is housekeeping genes or control genes. And in this example, as you can see, both control genes, both in the untreated and the treated case, they are pretty much the same. And that's what you would like to have. Now, how do I choose which is a good control gene and which is not a good control gene? I don't actually like this word housekeeping genes because they are not really housekeeping. They vary between tissues and they vary from one cell to the other sometimes. And classically used is something like GAPDH, but that's not necessarily a stable control. It depends very much on your assay whether you're looking at general any effect which would, for example, mess up your general gene expression pattern, then it can easily also affect any of your housekeeping genes. So general guidelines, again, would be expression levels must be similar in all tested samples. They don't necessarily have to be similar in all tissues in your organism or across organisms, but at least in the things you're testing, they should have a similar expression level. They should be resistant to the experimental condition. So if you see there's a big difference after you treat your samples, for example, then don't use it as a housekeeping gene. And finally, as we talked about before, you want primers which are giving the same kinetics, so the same efficiency for both your control and your target gene.
Within a certain range? Like above
So if about the efficiencies, they should all be in the 95 to 105 range. If you have two primers which both have 95, that's perfect. Then you're comparing the same range, although they don't amplify with 100% efficiency, they amplify with the same efficiency in both. Still, if you have two primer pairs which amplify with 80%, I still wouldn't go for it. It's just not a good qPCR result. Now we talked about GAPDH, and other commonly used housekeeping genes or control genes would be beta-actin, tubulin. Then oftentimes people use either rRNA directly or any transcript of ribosomal proteins. And they can be good controls in some cases, but then are not necessarily good controls. So if they work for your system, if you don't see any change ever and they're totally stable, fantastic, you can go for it. In case you don't, and that happens many times, you can do the so-called basket normalization, which is a lot more resistant to treatment, and it's a lot more accurate. And therefore, you use 3 different control genes, or however many you want to use, but 3 is generally what people do. And you use the geometric mean of those 3 control genes and use that for normalization of your target genes. And
So you would use the combination of those same 3, or?
You can use any 3 which you think are for your sample stable. There's usually, nowadays, there's a lot of publications out there which you can read up which genes are stable in which tissue. At least definitely that's the case for human, mouse, hamster, and so on.
I'm sure not likely for bees.
That I don't know. That I don't know. But there is a lot of publications out there where people checked which genes are expressed stably and accurately across tissues and across organisms. And you can definitely start from there and try some of these. And if you then see they are still varying, just choose a different one. So maybe you want to start with 5, which you test, and choose the best 3 of them.
Yeah, we've used FPL1.
Okay, if you have something that works well, don't bother.
Yeah, if you're,
But-
like, I was wondering, because people tell me different things. So if you have one that you know really is stable, it's rock
Yeah.
steady, you don't have to do the basket.
No,
You
no,
can
no,
do
no, no, no.
personal ones. Okay,
That's
I-
just,
under my
no, you don't
mind
have to
too.
do that. No, you don't have to do that. It's just, it's making it more robust and more accurate in case you're worried about your control genes. If you're perfectly fine and if it always comes at the same CT, whatever you do, you don't have to worry about it.
My intuition is often wrong, and so far it's here too. And I would guess that in an ideal world, your control gene would have a CT value, say, midway
Yes.
between your control and your treatment.
Yes, so it's always best to choose your control gene with a similar CT to your target gene. It should be at least in the same range of, let's say, 2 to 3 cycles in either direction, that's fine too. And obviously if you do a knocked out, you have like no target gene in one case or maybe 10% left, then you can't do that. But then you would go for the normal expression in normal tissue.
And an unrelated question
Mhm.
just occurred to me, this goes back several slides, but have you noticed differences in efficiency? It seems to me that the limiting step is the efficiency of your cDNA
Yeah, that's absolutely correct.
So
Mhm.
how do you validate
Mhm.
or estimate the relative?
That's a very, very good point. So of all these things, although the qPCR is actually quantitative, the cDNA synthesis is not at all. So what you absolutely have to do is a consistent amount of RNA as starting material. So you always have to check what's my RNA concentration and use the same amount of RNA for all of your reactions. You also have to be careful about the linear range of your cDNA synthesis reaction. They usually tell you that in the manual, this cDNA synthesis reaction is stable between 10 nanograms and 1,000 nanograms, let's say, and then you shouldn't use 5,000 nanograms because you're not going to have a linear amplification anymore. So that's certainly one thing if you're really worried, and that's also what I did during my PhD. I did a dilution row. So I actually made cDNA of 10, 100, 1,000 nanograms, or you name it, whatever you want to use, and checked if there is any difference if I use different amounts of RNA starting material, and what would be the optimal range for me for my assay. So if you really want to make it 100% correct, go for it and do a RNA dilution row first for your cDNA synthesis and see what's your stable linear range.
What's your quantification there?
So usually you're good with nanodrop, unless, so the range of nanodrop is between 10 nanograms per microliter and 2,000. So if it's a lot less or a lot more, you either dilute it or you have to use a different method, which would be bioanalyzer or qubit fluorescence based.
This is you're doing RNA or the cDNA?
For the RNA. For the cDNA, you cannot necessarily measure the concentration because you have your primers in there and your DNTPs which have not been used. So it's difficult to afterwards estimate how efficient it was. You can only really estimate by diluting your RNA and then running a qPCR. And I have only one more slide, and then I'm done. I just want to point out this paper because it will, I think, answer any residual questions which you might have. It's called the MIQE guidelines, or minimum information for publication of quantitative real-time PCR experiments. And it is a very extensive list, and you can tick the boxes of what you should do with your, how you should prepare your RNA, make your cDNA, which controls should you use for your qPCR reaction, which control genes would be appropriate, anything. And it's very extensive. I would not recommend to adhere to all of these points because otherwise you can make an entire paper just out of your qPCR reaction. But it is certainly helpful if you have any questions. It has a lot of good background material. And that's the last thing I wanted to show you today. If you have any additional questions, please feel free to ask me. Yes?
Actually, I do. So with this one that you just said, it's very extensive and complete, and you probably don't need to do all of that, and yet he's calling it the minimum.
Yeah.
And you're telling me it's not the minimum.
Trust me, if you read it, you'll understand what I mean.
So I can extrapolate to understand what you're saying, but I'm trying to figure out
what you actually should
the
do.
minimum is.
I think the minimum would be melting curve analysis, run your primers on the gel, make sure they are specific, make sure they are efficient, make sure that you're using the right control genes for your assay, so something which is stable, something which is accurate. If you can't find anything, use the basket normalization. That's the main points, I think. And there's a lot of more things which you can optimize, but if you adhere to these 3 points, you are in 99% of cases covered.
Okay.
Hey, do you have a reference for microRNA?
MicroRNA analysis?
For kind of, it's outside of microarrays to validate. Do you have a set of guidelines to assess micro?
Actually, there is for microRNAs.
MicroRNAs, kind of.
I don't
assessing
think
them
there
by-
is anything out there for microRNAs, as far as I know.
Yeah.
Like, there's no such guidelines as there is for standard long genes. As far as I know, microRNA people usually use northern blotting to verify that data. In some cases, you can also use qRT-PCR, but if I was working with small RNAs, I would definitely go for northern blot, although it's elaborate and not fun. But I think it has its justification there.
All right.
Additional
That was
questions?
fantastic. Let's thank Sarah.
Thank you for having me.